So how, your school, I mean, in terms of the COVID restrictions, so now, actually, we now start, all the instructor allowed to, basically take off the mask, but still, student need to, wear the mask during the class, starting, I think, 2 days ago.
Here, yeah, actually here there's a lot of frustration, I think, or at least I'm frustrated. Cornell has been a leader in dealing with the pandemic, and we've gotten a lot of national attention for that. But in the last week, the Ithaca City School District, where my 2 boys go to school, they've already announced they actually are allowing kids to go back to school starting yesterday with the option of not wearing a mask if they choose to take off their mask. Ithaca College, which is right next door, they announced their plans this week already. And Cornell has been totally silent. We have no direction from the university yet about whether we can take masks off or not. So yeah, so right now we're still continuing, everybody is wearing masks in buildings. And I'm fine if it stays that way, but I just wish they would come out and make an announcement one way or the other. But.
Right. How about, California?
I think we just had those announcements maybe 2 days ago.
I see.
You don't need to wear it in lab now.
I see.
If you choose not to, yeah.
I see. Actually, my school, wearing mask is the mandatory in the lab, but that is only applicable to instructor when they give, the lecture basically. So I mean, but what, nowadays, I wear mask because cold weather mask protect me in some sense. So anyway, okay, so I guess we probably want to start our start. Okay, so good morning, afternoon, evening, hola, hello, bonjour, ni hao, konnichiwa, 안녕하세요, and hello all. Welcome and thank you for joining us the 23rd seminar and Cornell Day. We are currently seeing the crazy war. How many more people should die after the 2 years of the pandemic? This madness must stop. And given that the energy is one of the selfish reasons for Russia's invasion of Ukraine, I would say that a clean energy is needed more than ever. Also, I hope that health issues, including COVID, should be addressed as soon as possible. Okay, it is my tremendous honor to introduce very briefly our pioneer speaker, Professor DeLisa from Cornell University. He is the director of the Cornell Institute of Biotechnology, and more importantly, he is one of the authors of the famous textbook called Bioprocess Engineering, Basic Concepts with the Dr. Shuler and Dr. Kargi, and both of them is the author as well. And I learned my biochemical engineering long time ago by reading that textbook and have been using that book for my own course. And if this meeting is in-person meeting, I would bring my book to get his signature. So Matt, this is my great honor to finally, meet you after long, time, we cannot meet in conference, and the virtual podium is all yours. Thank you so much.
Great, thank you, Taesuk, for the really generous and kind introduction, and for the invitation to be part of this really amazing seminar series. I'm gonna just take a minute here to share my screen, and please let me know if everybody can see that. Okay, great. So I do want to just take a minute and echo some of the words of, I know several of our previous speakers in commending Tae-Suk for putting together what I think is a really important and powerful seminar series. I think, so I'm a big believer in the creation of on-ramps for creating opportunities for individuals in all aspects of life and science and everything that we do. And I see this as a really critical on-ramp for especially our young aspiring colleagues, junior colleagues who are themselves aspiring to become faculty members and the pioneers that they get the 5 minutes here at the beginning to introduce themselves. So just a very warm gratitude to Tae-Suk for putting this together through a lot of challenges. I'm sure I've heard some of them, and I know it hasn't been easy. And so the perseverance is really greatly appreciated. So today, what I thought I would do is just very briefly introduce you to some of the work that my lab has been doing over the last, 20 years now at Cornell. And it's largely around a theme of understanding and engineering the protein machinery inside living cells for trying to solve complicated problems in biology and medicine. Before I do that, I also want to echo some of the remarks that Tae-Suk made. And I don't want to make this political, but I think it would be a missed opportunity to not make a comment. And what I do want to say is that I stand 100% behind the roughly 160 Nobel laureates and countless others who have come out and voiced support in various ways for all of the individuals in the Ukraine as it deals with this really, really horrible and challenging situation. So I think it's important to say that my thoughts and prayers are with anybody here today and around the world who may be suffering as a result of what's going on in the Ukraine. Not to continue with the downer messages here today, but I think another theme that has been echoed in many of the talks given by my colleagues through this seminar series is that humans and human life on this planet is threatened in ways that it has never been threatened before. And many of these threats are global in nature. So these challenges are not easy challenges to solve, not the kinds of challenges that one person or one technology can address. But the more that we think about how the work that we do can impact some of these challenges, the greater hope we will have to continuing life on the planet past the 21st century. So I won't belabor all of these challenges, but we know, for example, that life on this planet, humans have had a significant impact on climate. Estimates are that the temperature on Earth, surface temperature, has increased about 2 degrees since 1900, with the largest increases happening as recently as the 1970s. So very clear evidence that we are impacting the climate, and then this is having consequences that we are seeing on a fairly regular basis nowadays. It's also established that human-made material waste is obviously at an all-time high, and whether this be plastics or other waste materials that are predominantly not getting degraded or recycled is creating obviously another global scale challenge. Food supply and global food security continues to be a major challenge. When you think about a population of 8 billion people that is continuing to grow, how we provide food to that population is of growing concern, and one that needs to be thought about and dealt with very carefully. When you think about access to both drinking water, clean drinking water, and essential medicines, estimates are that about anywhere from about 1/4 to 1/3 of the world's population lack access to potable water and to the medicines that many of us take for granted. And then, of course, most recently is the emergence of a global scale pandemic threat with COVID, and concerns that there may be other COVID-like situations on the horizon as other viruses transition from their host into humans. And so all of these things, and there are others, and again, I don't mean to be a downer here this morning, but we have a lot of challenges in front of us. Another way to think about this, though, is that there are a lot of opportunities, right? These challenges create opportunities for creative scientists and engineers, especially the young aspiring faculty candidates that are the feature of this seminar series, that hopefully they will be thinking about and dealing with in their research programs as they get going and begin to have an impact, not only locally in the students and the work that they do, but also in the impact that they have on a global scale. So what my lab over the years has been doing, and the work, if I were to summarize in a single sentence what it's all about, is trying to leverage the biological machinery found inside of living cells in ways that can have an impact on some of these challenges. We don't work on all of the challenges that I just mentioned, but certainly with respect to essential medicines, with respect to pathogenic threats, this is an area where my lab has spent a lot of time thinking about how we might harness biological machinery to impact these problems. The work draws on a number of areas. Synthetic biology is one, but we also employ engineering principles. We employ metabolic engineering. We employ protein engineering, molecular biology, recombinant DNA technology. So many toolkits that we bring to bear on trying to tackle some of these really challenging problems. So in the interest of time, because I really want to make sure that I get off the stage quickly and leave Josh with ample time to tell us all about the exciting work that he's been pioneering. What I want to just do is tell you two very short, or give you two quick examples of the types of machinery that we have been trying to harness and the applications that arise from being able to harness that machinery. So in the first area in my group, and this is one that has grown in the last 10 years. It was not something that I started working on immediately as an assistant professor, but started later in my career. But it's an area that we largely have been referring to as synthetic glycobiology, which is the rational construction or intentional alteration of glycosylation pathways. So pathways that are involved in producing carbohydrate structures, and then in some cases, conjugating these structures either to lipids or to proteins. Taking these pathways and being able to address them using chemical and biological approaches in combination with metabolic pathway engineering and synthetic biology tools creates an opportunity to create designer glycomolecules. And so this is really important because glycomolecules have become and are being recognized more and more as a really critical part of basic biology, but also feature at the heart of many important application areas related to medicine, materials, and other areas as well. One example of the work that we've been doing by being able to harness cellular or cell-based protein glycosylation machinery is in the ability to democratize access to essential medicines that I just mentioned are limiting to large parts of our population. One way we've done this, although not the only way, is in the creation of a technology platform, a cell-free technology platform for producing conjugate vaccines, which are vaccines made of a carrier protein that's modified with an oligosaccharide antigen. This is work that our lab has been pursuing in very close collaboration with a friend and colleague of mine, Mike Jewett at Northwestern, and two of our students who themselves are rising stars, and hopefully we'll have an opportunity here to speak in the seminar series, both of them looking for faculty jobs in the very near future. So Jessica Stark, who's now a postdoc at Stanford, and Tommy Jaro and Tamichai, a grad student in my lab who is now a postdoc at University of Copenhagen. The two of them together teamed up to do some really powerful work on creating a cell-free platform for making glycoproteins. And they then leveraged that technology to turn it into an on-demand, portable, and low-cost biomanufacturing platform that's capable of making glycosylated vaccine carrier proteins. And we think this has tremendous opportunities and applications for point-of-care or decentralized production of these really important class of vaccines that are generally used against bacterial pathogens. And so work that we're very excited about, and again, emanates from our ability to understand and control protein machinery, in this case, the machinery involved in making glycoproteins. The second story that I'll just tell you quickly about that we're pursuing, or we have been pursuing over the last 5 to 10 years, is in the area of synthetic immunology, which we define as the rational design and construction of synthetic systems that can perform complex immunological functions. And so there are a lot of different ways one can think about synthetic immunology. The way we think about it, and we're not alone in this thought, is that there are tremendous opportunities for engineering microorganisms as modulators of the human immune system. This could be intact and living microorganisms, but it can also be components derived from those microorganisms. And my lab is actually working on both of those areas simultaneously. The example that I want to share with you is in using derivatives of microorganisms known as outer membrane vesicles. So we now know there's a greater appreciation that all living cells on this planet have some unique mechanisms for releasing portions of their outer membrane in the form of nanometer scale vesicles. And bacteria were some of the first where this was recognized. We now know this is also a mechanism that occurs in higher organisms as well, including in human cells. In bacteria, the release of these outer membrane vesicles creates an incredible opportunity to create outer membrane mimics that can be used to deliver vaccine antigens directly to the immune system. And so we've been doing this over the last decade or so, starting first with protein antigens, and then actually marrying work in OMV, or outer membrane vesicle vaccine production, with the synthetic glycobiology that I just told you about, to create designer glycosylated outer membrane vesicles as an alternative form of a conjugate vaccine. What we're most excited about here is a number of things with respect to how these vaccines are made and how they work. One thing that's really powerful about them is that the OMV structure itself, the ultrastructure, presents antigens in a way that favors B cell activation. And we think this is because of the network of biomolecules that you can put on the surface can promote BCR clustering and other events that are really critical for getting potent immune responses. They are able to stimulate innate and adaptive immunity. So there's a potent and broad immune response, turning on both arms of the immune system. We also know that the OMVs themselves have intrinsic adjuvant properties, meaning that they promote an immune response to an antigen that is stronger than you would otherwise get with the antigen alone. And for these reasons, these OMVs are, in essence, an all-in-one antigen, adjuvant, and delivery system that has really incredible potential for both vaccine development and creating immunity against infectious agents, including bacteria and viruses. But also, more recently, we've been using these as a research reagent to help us in the discovery of very high affinity antibodies that are specific to the antigens that we engineer and display on the surface of these particles. So the last slide here, and then I will happily turn the stage over to Josh. But I think this is probably the most important slide. And it's an acknowledgement slide like we always put at the end of our talks, but it's a little bit different. It's different because what I want to say, and perhaps the most important thing I'll say here this morning, is that for a seminar series focused on young investigators, I think it's always important to remember how we got where we are, who helped us get there, and to make sure that we give the due credit for all of that. So I left this slide clean of funding acknowledgements. I do want to also acknowledge funding agencies that have helped us along the way. But the people that have made the work that my lab has done and have helped me get where I am in my career, just a few of them are shown in this slide. And I do want to apologize for the numerous individuals that I couldn't fit onto this slide. But let me just say that I've been really lucky to work with an amazing group of postdocs, grad students, and undergraduates. In fact, Jessica Stark, who I mentioned a few slides ago, who was a grad student in Mike Jewett's lab, now a postdoc in Carolyn Bertozzi's lab, was an undergrad at Cornell, first and foremost, and worked in our lab. So it's a very small world and a really lucky opportunity to work with some incredible people. And I wouldn't be here if it weren't for the efforts of all of these amazing people, both past as well as the present members of my group who are shown here in this slide back in 2021 when we first could get together outside with masks off. It was a really special day. I also, though, wanted to take a moment to recognize and honor two of the most important people in my academic life, my PhD advisor, Bill Bentley, and my postdoc advisor, George Georgiou. Again, two individuals, if it were not for them, I certainly would not be where I am today. And it's not just that they helped me get to my faculty job, but they continue to this day to play a very important role in helping me with anything in my career, including award nominations and just opening doors for me that would have remained closed if it weren't for them. And then lastly, for the young and aspiring faculty members in the seminar series, one thing to consider too is that once you start a faculty job and you are in your independent career, you're still not independent, and you do still need a lot of help. And I was lucky in the start of my independent career, arriving at Cornell with really no idea how to do anything that a professor needs to do. I was really lucky to be helped and mentored by two amazing individuals. Kelvin Lee, who was my direct faculty mentor, who was assigned to be my mentor, but went above and beyond the call of duty and helped me in every way imaginable. And then, of course, Mike Shuler, who was not my official mentor, but mentored me informally in so many different ways. And as Tae-Suk mentioned, one of the greatest honors I had was when Mike, a number of years ago, asked me to help him revise the Bioprocess Engineering textbook to the 3rd edition. That book also had an incredible influence on my own career. I was a student who took Bioprocess Engineering from Bill Bentley, read that book cover to cover, and actually, shortly after arriving at Cornell, I did ask Mike Shuler to sign my copy of the book. So I do have a signed copy of the book. I haven't been able to get Varkey's signature yet. I'm working on it. And so anyway, I'll just wrap up here. And again, I just want to thank Tae-Suk for the chance to have a few minutes here to share with you some of the stuff that my lab is doing, but also to be a part of this really incredible program that I hope continues on as long as possible. I think it's a really incredible and important on-ramp that we need to continue to support. So Tae-Suk, thank you. And Josh, I really look forward to hearing your talk.
Thank you so much. Amazing talk. And also all the people I also love, especially, Professor George Georgiou. Actually, I almost joined his lab unless I joined the Chris Voigt lab. Also, I echo everything you mentioned. I still, respect, George Georgiou, and also, my own advisor. Without them, my career would not be possible right now. So thank you so much in this amazing talk. And then I completely agree with you. All global issues, you mentioned earlier, should be addressed as soon as possible and will be addressed by synthetic biology and other technologies if we work together, not by having crazy wars and challenges are our opportunity, because we have our future leaders like young speakers and audience in this seminar series worldwide, including today's speaker, Josh. So this is my great honor to have both of you and all the audience. And thank you, everyone. This is my great privilege to continue until I die. So, okay, so now the main speaker of today with a longer introduction. So Josh is a PhD candidate in genetics at Stanford University. In fact, he is the first PhD candidate, brave, I would say, who is giving a talk in this seminar series. Actually, there are some more, PhD student, but he is the first because the last 6 months have been either postdoc or faculty members, but I really appreciate he volunteer kind of doing earlier. Also, his research aims to systematically define the proteins and DNA elements that control human gene expression by developing new genetic screening technology in the labs of Dr. Bintu. She's also, junior faculty, and Dr. Basic. And Josh's research is supported by the NSF GRFP, one of the, great kind of award to the graduate student. And previously, Josh helped develop gene therapy using CRISPR genome editing at a biotechnology startup company. And actually, I met him last year virtually when he gave brilliant talk. And then it's my great pleasure to have him again. And he served as a mentor to Stanford iGEM and is a project lead at the Stanford Genetics Advocacy Committee. That means he also cares about younger generation. And that means he is true future leader. And he earned a bachelor degree in biological mathematics from University of Pennsylvania. And what else do you want to know? In true rising star, and I believe all the people Matt mentioned, if they are listen to this talk, he should be higher because competition will be very, very high. So Josh, take it away. Thank you so much.
Thank you, Taysek. I'll just share some slides. So I really appreciate this opportunity and the energy and enthusiasm you've brought to creating this seminar series. I think it's, solving for a pretty important problem with kind of the relative isolation of the younger scientists during the pandemic with fewer opportunities to go to things outside of our universities and meet people like you. So yeah, really appreciate you organizing this and feel very inspired about the potential huge impact that some of this research can have, seeing Matt's introduction talk there. And hope to ask you some more questions about that synthetic glycobiology in the future, because that is really fascinating. So thanks so much for this opportunity. I will look forward to telling you all about our project to systematically discover and characterize the human transcriptional effectors. So this has been a collaborative project between my co-advisors at Stanford University, which are Dr. Michael Basic and Dr. Lakbar Bintu. So I wanted to get started with this quote from a biologist turned anthropologist named Donna Haraway. So about 30 years ago, Donna wrote that in modern biology, the organism has been translated into problems of genetic coding and readout. In a sense, organisms have ceased to exist as objects of knowledge, giving way to biotic components or special kinds of information processing devices. I thought this is a kind of fascinating and apt description of the way that biology has been going, especially synthetic and systems biology, and how kind of ahead of her time she was in describing things this way. It's only over a decade later that we have really kind of the main systems biology textbook coming out, where we see this idea of the organism as an information processing device really being kind of put into practice with this model on the right, which kind of shows information from an environment coming in, interacting with some transcription factors or proteins in an organism and controlling the genes in the genome of that organism, which in turn can affect its environment. And this has been the simplified model is definitely missing almost all of the details, but I think it is still fascinating and gives us kind of a handle for asking some fundamental questions from the perspective of a systems or a synthetic biologist. And so here I'm just adapting this model to make it kind of more tailored to be the human cell. So that means just putting a nucleus in there. And we know a few of these numbers in a human cell. We know that there's about 2,000 of these transcription factors, and we know that they have to control about 20,000 genes. So in the main project that I was going to focus on today, we've really been studying how these endogenous transcription factors, as well as synthetic transcription factors that we can add into the cell, how these kind of come together to control these endogenous genes, as well as synthetic genes that we can add into the cell. And if you just kind of look at this part of the model, I think there's like a few questions you might have about how does that transcription factor interact with that gene. So for each TF, we would like to know, of course, what is its target gene, and I would say that's been relatively well studied with methods such as ChIP-seq and groups such as the ENCODE consortium. But we don't just want to know the target gene; we also want to know something about what is the relationship that's represented by this interaction. Is it activating that target gene? Is it repressing that target gene? What is the kind of quantitative information, the relative strength of that interaction? And is there potentially in some cases conditionality where it's only an activator given certain contexts such as the cell type? So this might be a simple enough problem if there were only a handful of these transcription factors, but part of the big challenge of biology, of course, is the complexity and scale. So we know that there are about 2,000 of these. So this gives us a big motivation to come up with new methods that allow us to kind of tackle this problem in a more efficient or parallelized way. So zooming in a little more on how these transcription factors look. We know that there are thousands of these in the human nucleus, and they have at least two kind of main functional domains. There has to be a DNA binding or way for that transcription factor to localize to its target genes. And then there is a effector domain, which is really the more less characterized part that I want to focus on today. And these effector domains are the part of the protein that actually interact with other kind of cofactors, such as co-repressors or co-activators, and ultimately upregulate or downregulate the expression of their target genes. So hopefully you're curious to hear more about effector domains, but I do also want to mention that these could be useful as well for building new gene regulation tools and technologies. I think there's kind of a nice cycle between as we make more measurements of these kind of protein components, like effector domains, we then get better at using them as tools to go and manipulate the system. So first of all, for research purposes, perhaps many of us have heard of CRISPR interference or CRISPR activation, kind of research tools for controlling the expressions of genes in cells, and as well as gene therapy. So we can imagine that potentially one day in the future, we will be treating diseases with gene therapies that are not just kind of one shot and done with a gene that's expressed on full blast for the rest of your life, but that are more regulated as we get better at building kind of synthetic transcription factors to control the expression of these therapeutic genes in useful ways so that we get the right dose of the right gene product in the right cell at the right time. Okay, so we kind of set out to study these effector domains. And I think the main kind of trigger for starting this project was the realization that these domains in these proteins are finally applicable for studying with high throughput genetic libraries, because these DNA libraries that we use to kind of encode synthetic gene products had finally gotten longer, now reaching up to 300 base pairs long, which is finally long enough for us to actually start encoding full protein domains, up to about 80 amino acids. And if we can operate within that synthesis limit, we can capture at least around 70% of the known annotated domains that exist in human nuclear proteins. So this represented an exciting kind of opportunity to just build a library of all of the putative possible effector domains coming from nuclear proteins in the human proteome. And so that's kind of what we did. And we test these pooled protein domain libraries with a new method that we're calling high throughput recruitment, or HT recruit. So here we have a library of all of the annotated PFAM domains from human nuclear proteins cloned as fusions onto the R-TET-R DNA binding domain. So we can deliver these constructs to cells with a lentivirus at a low dose, such that each of these cells expresses one of these domain elements. And these cells are engineered reporter cells, where they have this reporter gene that has two parts, a citrine fluorescence marker and a synthetic surface marker. And then just upstream of that, there's this TET-O binding site. And this is where we can recruit the R-TET-R DNA binding domain to come down and bind to the genome, bringing in this putative effector domain into proximity with this promoter. And so the way this experiment might look is, in the beginning, the cells start out on, so we see the high citrine, and then we can add doxycycline, this inducer molecule. And for each of the cells that happens to have received a domain that actually works as a repressor, this target gene will be silenced over the course of 5 days. And then we can take a time point and make a measurement, or we can wash out the doxycycline and look at subsequent time points. Does that cell stay silenced with a permanent epigenetic memory, or is gene expression reactivated over time? And then at each of these time points, what we do is we mix these cells with magnetic beads, which can bind onto the synthetic surface marker component that we engineered. And so this gives us an easy way to separate the cells that are on, that bind onto the beads, versus the cells that are silenced or off. And this is just a more kind of scalable method than cell sorting. And we then sequence the domains in those two populations and compute an off to on enrichment for each of these domains, which kind of gives us a score for how well each domain worked as a gene expression repressor. And I want to point out that this has been kind of an exciting field over the past few years, with several related methods being published by a bunch of great labs, including in other organisms, such as like Drosophila and yeast, which has been very cool to see. So digging into the results a little bit, well, I first want to point out that this has been not only a collaboration with my advisors, the Bassick and Vintu labs, but also with many trainees in both of these labs. Really, we had to kind of come together and try many different things to kind of get to the point where this method can work well and reproducibly. We see a good correlation between biological replicates. And we can see with high off to on ratios, these repressors coming through in the upper right corner. And it was good to see that these include some previously known repressor domains, such as, for example, the chromoshadow, as well as some domains of unknown function, or DUFs, which we were excited that we can nominate gene repression as an initial potential function for these domains. And also, I'm not showing this, but want to mention that we do a counter screen where we measure the flag tag based expression level for each of these domains in cells, so that we can filter the data to focus our analysis in the domains that are well expressed in these cells. So To zoom in on the largest family of repressor domains in our data, this is the crab family. There's over 300 of these that come from different human transcription factors. And we find that 92% of them function as repressors. A handful of these have been previously characterized with low throughput methods in human cells and have been shown to bind to CAP1, which is the crab associated protein 1, which is a co-repressor, which in turn can recruit histone modifiers that can methylate the chromatin of this target gene and silence gene expression. And somewhat famously, the CRISPR interference system is really DCAS9 fused onto the crab repressor that comes from the zinc finger 10 gene in particular. So one of the questions for us is why do some of these work as repressors and some of them do not? So what I looked at here is a comparison of our data with co-IP mass spec results from full-length crab zinc finger proteins. What we can see is that when the full-length protein does not interact with CAP1, in our data, then the crab domain does not work as a repressor. Whereas for the full-length genes that do interact with CAP1, the crab domain is a repressor with this one kind of outlier here, which was actually poorly expressed in our cell type, which could explain that one difference. And then we also looked at this by using CHIP exo sequencing data to look where do these crab zinc finger transcription factors actually localize on the human genome in relationship to this co-repressor CAP1. We could see that if the TF includes a crab that is a repressor in our hands, then they co-localize on the genome with CAP1, and whereas the non-hit, non-repressive crab domains do not co-localize with CAP1. And then most interestingly, we saw this kind of trend wherein the younger, more recently evolved crab domains are the repressors, and these rare crab domains that do not work as repressors are actually the more ancient crab domains. So this left a lot of open questions for us, of course. Well, then what are these ancient crab domains actually doing in human cells? Which I will get to in just a few minutes. But first, I wanted to mention that we can also use HT-Recruit to do deep mutational scanning. And so here we did a deep mutational scan of that zinc finger 10 crab domain, the repressor that's used in the CRISPR interference system. And what we have here is the full-length sequence of that crab domain. And we're doing a mutational scan where we make every possible substitution at every possible position and score its repression activity. And you can see that there, in some residues, there's these dark red stripes. So these would be residues where most mutations are null mutations that break the function of this effector. And the pattern is very clear when we make larger triple mutations that this A box portion of the domain is really critical for its gene silencing function. If we then map those significant residues onto the structure of this crab domain, we can see that they are similarly oriented in 3D space on this kind of helical turns of this domain. And it turns out that these correspond well with residues that were shown to be important for CAP1 binding in previously published in vitro recombinant protein binding assays. We can get a fuller sense of the picture by using those additional time points to look at the epigenetic memory of this crab silencing effector. So again, at day 5, we found the A box portion was critical for silencing. But if we look at later time points, we can see this B box portion actually, while not being necessary for silencing, it does contribute to the overall silencing strength. And then interestingly, at the latest time point, we can see these dark blue patches. So these are mutations in the N-terminal region of the effector that actually enhance its function above wild type. And so it turns out that this N-terminal portion is very poorly conserved and is in fact uniquely found in the zinc finger 10 crab domain and no other crab domains, and is actually spliced on from a separate exon than the crab domain. And what we found is that by making mutations in that poorly conserved region, we can stabilize the domain and thus get a stronger, an enhanced kind of function in our assay. And this is validated here, where we can see, for example, with the wild type, over the course of 5 days of treatment, we get in full silencing of a population of cells. And when we release the recruitment, some of those cells reactivate while a minority of the cells stay silenced, and we can kind of enhance that function by mutating the N-terminus. So making mutations in a known kind of component like the zinc finger 10 crab domain is one way to potentially improve our tools like CRISPR interference, but another way that I would put forward is to go back out into nature and identify new paralogs that might have enhanced functions. And so what we looked at here is if we look across all of the different crab repressor paralogs, how do they kind of rank in these screens? And here we're doing HT recruit in multiple cell types, targeting multiple promoters, and looking across these different contexts, how do these crab repressors kind of rank? And we can see that the zinc finger 10 crab does not rank that well. It's often around number 200 out of the list, whereas some of these other ones are much more consistently ranked near the top, such as the ZNF 705F. And then I want to point out that also another group working on similar methods recently published that the ZIM3 crab domain is an improved paralog over the zinc finger 10. I think that's corroborated as well by our results. So if we actually put these repressors onto DCAS9 and see how well do they work when we target an endogenous human gene like CD43, we can see that in the unshaded light gray is the negative control where the cells start out on. And then in the shaded color, we see that the cells shift to the left, or they get silenced when we add in this crab repressor and a guide RNA. And this repression is pretty good with our enhanced version of zinc finger 10, but we can make it even kind of more homogeneous with a clean, narrower peak if we use a stronger paralog of the crab domain. And this turns out to be kind of consistent across hundreds of guide RNAs targeting about 50 different genes, where we see this enhancement of the effects where we're getting greater phenotypes in the screen if we use a new paralog for the guide RNAs that work, while there is going to always be this minority population of guide RNAs that are potentially bad choices of guide RNAs that don't have much of a function. So the second largest family of repressor domains in our results was the homeodomains. So homeodomains are found most famously in the Hox genes, which are these master regulators of development. And Hox genes are really interesting because they have this kind of fascinating correlation between their organization in the genome and the spatiotemporal dynamics of their expression in the developing embryo. So Hox genes are found in clusters in the genome, where these 4 different clusters where you have up to 13 Hox genes all next to each other. And they are all similarly oriented or transcribed in the same direction. And we can kind of number these paralogs from the 3' end of the cluster with the lower numbers, starting with 1 up to 13 at the 5' end of the cluster. And then it turns out that if you look at where these genes are expressed in the developing embryo, the 3' Hox genes are expressed early at the anterior end of the organism, whereas the 5' genes are expressed late at the posterior end of the organism. So this is kind of called a collinearity between their genetic organization and their expression pattern. And what we were excited to see is there's an additional collinearity between their genetic organization and actually the strength of their homeodomain function as repressors, where in the 3' or early Hox genes, do not have strong repression function from the homeodomains, and it gets stronger as we get to the 5' end, or these later expressed Hox genes. So it's interesting to think if this might be related to the so-called posterior prevalence rule. This is a rule that states that when you have multiple Hox genes expressed in the cell, the more 5' Hox gene tends to have a dominant effect on the phenotype of that cell. And so now we are curious if perhaps part of the mechanism behind this posterior prevalence is that the more 5' Hox genes are stronger repressors, and there might be some autoregulation in this locus where they can actually repress one another. So far I've talked about repressors, but we can also use H2 Recruit to study activators. And so we simply slightly change our reporter gene. So instead of a strong promoter, we're using a weak minimum CMV promoter here. So the cells start out off, and if we recruit in an activator, that'll turn the cells to on. And so this will give us a highly negative log2 off to on ratio with activators in this lower left-hand corner. And we're pleased to see some positive controls, such as previously known activator domains, come up in this assay, as well as, interestingly, the strongest activator was actually the zinc finger 473 crab domain. So this is one of those ancient crab domains that does not work as a repressor. It's highly divergent in its sequence from the consensus of the repressive crab domains, and with more acidic residues, which is a biochemical characteristic of activators. And so one thing we've been working on more recently is porting these new effectors onto CRISPR-A systems. And so CRISPR-A has previously been built using activators from viruses such as VP64 or this tripartite VPR activator. And what we can see is that when you target these to an endogenous gene, such as the surface marker CD2, which starts out off, if you recruit in a CRISPR-A system, we can shift that to the right and turn that gene on. And what we wanted to see is how do these new effectors kind of compare? And so if we look at just 3 of these new effectors by themselves, we see as singletons, they're really kind of quite weak activators of this endogenous gene. But if we start combining them, such as a pair, so N plus Z would be NCOA3 plus ZNF473, we get a nice kind of synergistic or more than additive effect of the combination. And this continues if we add all 3 together to create this NFZ tripartite activator, which nicely is still coming from human components and is still relatively compact at 145 amino acids. So what we found most exciting about this new combination as a CRISPR-A system is that it has a good balance of potent activation while still being easy to deliver to cells. And so here we're doing delivery using a lentivirus and marking the delivery with a BFP. And so we can see that with NFZ, we have a large population of BFP positive cells that are shifted over to the right because they're being strongly activated at the target gene. Whereas with VPR, we have a problem with our lentiviral delivery where there's very few BFP positive cells that we can actually measure activation in. And so to kind of show you how these plots relate, to make these histograms on the left, we have to gate for BFP positive cells. And in fact, there's very few cells that we're actually gating for because of the difficulties of delivering this larger viral activator system compared to the new one, NFC. So another application of new activators like this NFC could be for controlling the expression of therapeutic genes. So in the future, you could imagine a form of kind of safer gene therapy where the expression of a gene is tuned by simply taking a pill. So this has been kind of a vision in the field for a long time, where the idea is to use rapamycin to induce the dimerization between a DNA binding domain, zinc finger, and an activator domain to drive the expression of a cargo gene. But the previous versions of this kind of mini circuit relied on much larger activator domain called P65HSF1, and ultimately left very little space within this kind of delivery vector for the actual cargo gene, most of the space being taken up by the kind of transcription cassette. So by using this more compact NFZ, we can kind of simplify and shrink this transcription cassette, more than doubling the amount of cargo capacity, such that it's now around 2.3 kilobases, which would be enough for monoclonal antibodies, cytokines, and various other therapeutic genes we might like to deliver. And in kind of initial proofs of concept of this design, we can see that indeed it is inducible when we add the small molecule. And nicely with the new activators, we see less leakiness where there isn't, we're not seeing expression before we add rapamycin. Whereas with the previous system in our hands, we do see some leakiness. So far I talked about studying domains that come from this kind of PFAM annotated database of protein domains. We can also use these high throughput methods to discover unannotated functional effector domains in large proteins. So here's an example, which is MGA. It's about 3,000 amino acids long, and it previously had only 2 annotated domains shown in gray, both of which are involved in its DNA binding function. So what we did here is tile across the full length of the sequence in 80 amino acid tiles, shifting over by 10 amino acid windows. And then we can see these peaks, which are, these peaks are tiles that actually function as repressors. So which we're calling repressor 1 and repressor 2 here. And we can see that these validate when we study them individually, where both of these can work as repressors, although they do so with different dynamics. And then we can also minimize down from 80 amino acids by looking at what is the shared sequence between all of the tiles within this peak and testing just, in this case, 10 amino acids. We can see that that's actually sufficient for repression in this assay. So it's one thing to kind of find components that function as repressors, but what we would like to increasingly do moving forward is better understand the dependencies or the mechanisms for these different repressors. So the method I've been kind of developing to better understand how these repressors work is CRISPR screens, where now what we're doing is basically making a diversity of different genetic backgrounds in these reporter cells, where each of the cells is going to have one gene knocked out, and then repeating this recruitment assay, and in this case, sequencing the guide RNAs to measure what is kind of the effect of that knockout on the function of our repressor. And so when we do these types of CRISPR screens, we'll get a score, a significant score for each gene out the other side. And so if we do a CRISPR screen where we're recruiting the CRAB repressor, we see really high scores for its cofactor, CAP1, and for the chromatin modifier, which is recruited by CAP1, which is called SETDB1. So this is exactly what we expect for the CRAB repressor based on previous literature. But then what was kind of exciting to see is if we repeat this assay for MGA, for this tiny 10 amino acid new repressor domain that we discovered, we see that there are some shared dependencies like SETDB1, but then there are also unique dependencies, such as KDM5C, which is a different chromatin modifier enzyme, and actually MGA itself, suggesting that potentially this tiny effector domain actually works as a dimerizing interface with the full-length endogenous MGA. And lastly, we're interested to understand better how effector function depends on the biological context. So context could mean the cell type. You can imagine different cell types are going to express different cofactors to different degrees, as well as the target genomic locus. So you can imagine the chromatin state at different targets could affect how well these effectors can function at those targets. So we've been mapping effector function across target and cell type contexts. So what we have here is effectors along the x-axis, and then on the y-axis we have different types of contexts. So we've tried 2 cell types, K562 and HEC293Ts, a variety of promoters. Some of these are strong promoters that can be repressed, and some of these are weak promoters which can more easily be activated. And then we're recruiting either with rTET-R, where we can target kind of synthetic reporter genes, or with dCas9, where we can recruit to target endogenous genes. And so this kind of mapping, I think it's useful, I guess as previously mentioned, for finding tools like the best CRAB paralog that works most consistently across many contexts, but also for potentially picking up on interesting new layers of gene regulation. For example, in cases where we see high context specificity, wherein some effectors can work differently, for example, working as activators in one context and as repressors in another context. So to look at just 2 examples from here, we can see that while many effectors are kind of consistent across different recruitment contexts, there are some interesting cases where effector function does depend on the recruitment context. So first of all, we sometimes see cell type specificity. So here we're recruiting activators in K562 on the y-axis and in HEC293Ts on the x-axis. We can see there are some shared hits, such as the N, F, and Z strong activator domains that we're using in our new CRISPR-A. But then there are many activators that are really only working in K562. And we can see that in these individual validations here. So these positive controls like BP64 or FOXO, consistent in both cell types, but this WW domain, for example, is a very strong activator in K562, while giving us really no signal in this HEC293T cell type. And then most surprisingly, in some cases, we see actually opposite functions, wherein an effector can work as a repressor in one context and work as an activator in another context. So here we're looking at just the HLH family of effector domains. This is the 3rd largest family of effector domains. And we can see that there's this opposite function where it's actually inversely correlated how strongly they repress this PEF1 alpha promoter with how strongly they activate this CD2 gene. And while that's the case for the overall family, if we kind of zoom in on the subfamilies of this HLH family, we see that actually there are subfamilies that work in kind of coordinated ways. So the ID 1, 2, and 3 domains are really representative of this kind of opposite function as repressors and activators. The HEY1, HEY2 subfamily are functioning as repressors, but not as activators. And the HAND1, HAND2 subfamily are activators, but not repressors. So I'm going to wrap up there just to summarize some of what I spoke about. We've been using HT-Recruit to measure the effector function at scale, including using dCas9-based recruitment of these kind of fusion libraries to target endogenous genes and see how much each of these effectors can activate or silence gene expression. We can do this using known kind of annotated PFAM domains, but we can also do unbiased tiling of large proteins in order to discover where effector domains might be lying in the unannotated regions of these proteins. And we can also use HT-Recruit for deep mutational scanning, which helps us kind of map the sequence of this domain to its function, identifying the residues that are actually necessary for its function, and in some cases identifying enhancing mutations. And lastly, we are optimistic that these results could be a resource for people hoping to do synthetic biology. So we have, compact domains that are sourced from human components and are, in some cases, easier to deliver or stronger than the previously used tools. And so we hope these will be useful for perturbing transcription or manipulating the epigenome. So hopefully I've kind of convinced you that the picture for how these transcription factors regulate these genes is interesting and kind of diverse. And there are, cases where some transcription factors are very consistent, working strongly the same way on every gene that they target. There are surprising cases where these effectors work as activators of one gene and repressors of another gene. Cases where they might be conditional, working as an activator only depending on the cell type. And so with that, I'm going to thank all of you for listening. And just to give a few acknowledgements, this has been a really awesome collaboration between my co-advisors, Lachlan Bintu and Mike Bassick. And I've also worked really closely with many other students in their labs, such as Mike Van, Nicole, Galen was a postdoc in Mike's lab who really helped train me and is now faculty. And Aradhana has been a huge contributor. I also want to thank funding from the NSF and the NIH for fellowships and many other collaborators. And yeah, thanks again for having me and letting me tell you about that work. Appreciate it.
Wow, fantastic talk. That's really great work. I mean, I still cannot believe you are still a graduate student. So this is amazing. So any question from audience? Let me check the here. If let me see. So Q&A, actually nothing yet. And then no chat. And then you could raise your hand if you want to speak. Otherwise, okay, let me start my question. So, so compared to bacterial systems, I'm still in a bacteria guy. Your system have, of course, the many unknowns to answer. So again, the such challenges are our opportunities. And my question is, what are the critical remaining challenges in your field? And then if you have limited resources and time, which one challenge do we should tackle next decade?
Well, I think to start with one on the kind of applied side of this field, I think one thing that we would still like to be able to do is find tools where we can transiently target a gene and permanently activate it. This could be really important for treating some diseases. And we actually have the ability to kind of epigenetically modify and permanently silence genes, but not to activate them yet. It turns out that this is not only applied questioning because it actually comes down to we need to understand better activating chromatin modifications and what tools might already have evolved in mammalian systems to achieve kind of long-term activation of genes. So I think that'll be one area where actually some more kind of fundamental study on chromatin biology could lead to an interesting breakthrough for applications. Yeah.
I see. I recently, very interested in this in high order organism, and then I actually start to do some collaboration, and that many people actually talking about similar things. So this is probably the exciting time for the field. I guess. So let me check again. I guess we right now because right now my time is 11:10. Probably we want to close and then open up the real discussion. So let me close. So thank all for joining again and staying today. And we will meet again next week on March 10, Thursday, again, the same time, the same Zoom link. We'll have tentatively, Professor Chris Boyd from MIT, my postdoc advisor, and the founding editor-in-chief of ACS Synthetic Biology. So my academic dad, and or another, we kind of still kind of scheduling because of his kind of conflict, just recently kind of, arise. And also Feng Chao Song from UC Berkeley, Adam Arkin Lab. So my academic uncle, because Chris Boyd did his postdoc in Arkin Lab, and I'm also his official mentor through AICG mentor-mentee program. And as usual, the follow-up informal chat will occur without recording. So please stay here if you are interested in chatting with him and me, and I'll promote you to panelists who can speak and show your handsome and pretty face. And I'll stop recording now. And thank you so much for everyone, staying here. Okay, let me stop recording. In that way, Josh, feel better, I guess.